Okay, so good evening to one and all present here. Welcome to the course on Introduction to Complex Biological Systems. I am Srijan Sen, and today I will be discussing the week 11 live session, right? So I'm sharing my slides with you all. So I hope my screen is visible, right? So today's class is mostly it will deal with genetic engineering, that is impact on the human well-being and the details of it. So if we start, we will have to go around with the introduction to genetic engineering. Then we will majorly discuss about the recombinant DNA technology, the tools, the enzymes, vectors, and the basic techniques that are actually involved in this genetic engineering process. So starting with, so if we know that genetic engineers have developed this recombination technique to manipulate the gene sequences in plants, animals, and other organisms such that they can produce some recombinant organisms that have better effectivity, that are better effective than the original or the wild type constructs. So based on this, genetic engineering was taken up. So the desired DNA is cleaved from, say, a donating chromosome and of one organism, and then it is incorporated into the other ones using some restriction enzymes, stuffs like that. Okay, so the desired DNA fragment is inserted into a vector, usually that's a plasmid, and when the uh, using some ligase enzymes and some joining enzymes, so we call them as the DNA ligase, we create a recombinant DNA, and then you put that into another bacterial cell in the form of a plasmid, and you let it grow to create a gene library. Fine. So there are different historical ways of how gene has been manipulated. So in the later part of the 20th century, the term gene or the test tube babies, this particular term came to refer more specifically to methods of the recombinant DNA technology, gene cloning. So in which the DNA molecules from one or two sources are combined either within the cell or in vitro and then inserted into the host organism through which they are able to propagate. So we see that the possibility of recombinant DNA technology emerged with the discovery of restriction enzymes in 1968 by the Swiss microbiologist Werner Arber. So what is basically, so this is the most important part. So what is recombinant DNA technology? It is joining together of DNA molecules from two different species that are inserted into a host organism to produce new genetic combinations that are of value to science, medicine, agriculture, different biotechnological applications. And to focus of the genetics is the gene. The fundamental goal of laboratory genetics is to isolate, characterize, and manipulate the genes. Although it is relatively easy to isolate, sometimes you face quite a lot of difficulties in doing that. And what is important is that the wild type or the construct that's originally available might not have a better activity than compared to the recombinant gene. So, right. So what we have, what we do in a recombinant DNA technology, if I speak about that, so we have our donor DNA and we have vectors. So the donor DNA is amplified using different DNTPs. Then the different buffer system using a DNA polymerase, you amplify it, and then using the restriction enzymes. So there are certain restriction sites that are present in these sequences. So using the restriction enzymes, you first undergo restriction digestion of the vector as well as the insert, and then using the DNA ligase, you have to join them, right? And then what you do, you introduce this into the bacterial cell. So, so this is basically the recombinant DNA technology where you are creating multiple recombinant DNA sequences or libraries present in this. Uh, you can say about a different construct or say one particular protein, things like that, and you're inserting it into the bacterial cell for the expression of the protein. Now, there are certain tools for genetic engineering. We talk about the ligases. So, mainly in molecular biology, we know that ligases are molecular glues or molecular paste. So, the action of the ligase is opposite to that of a restriction endonuclease. Now, in the cell, the function of a DNA ligase is to repair single-stranded breaks, discontinuities that arise in the double-stranded DNA molecules during DNA replication. Now, DNA ligases from most of the organisms can also join together to different fragments of double-stranded DNA. Now, ligase catalyzes the ligation or joining of nucleic acid fragments by forming a phosphodiester bond between the directly adjacent 3' to 5' and 5' to 3' terminal. So, right. Now, there can be different types of ligation. So, first one can be you can use nick sealing, which we basically do. The other part is that you can join it using the sticky ended molecules, that is also done using the DNA ligase. So, once you can stick them, you are actually getting a recombinant DNA molecule or a plasmid. Now, the ligation reaction shows that the two blunted fragments join together. Now, although the reaction can be carried out in a test tube, it's not that very efficient. This is because the ligase is unable to catch hold of the molecule to be ligated and has to wait for the chance associations to bring the ends together. So, therefore, the ligation of complementary sticky ends is much more efficient. This is because the sticky ends will help in the complementarity of the base pairs of the cut ends, and that will help in sticking together. Also, you need a DNA ligase for that as well. So the different types of DNA ligases that are available is the E. coli ligase, which catalyzes the cohesive and the blunt end ligation in the presence of NAD as a coenzyme. You have the bacteriophage T4 ligase, which catalyzes the ligation of the cohesive ends, blunt end, DNA RNA hybrid and RNA RNA hybrids in the presence of ATP as coenzyme. And then you have the Taq DNA ligase, that's obtained by cloning the Taq gene into E. coli. So what are the uses? So to complement lagging strand synthesis during replication, then to ligate vectors, and foreign gene in the eDNA technology, to ligate linkers or adapters, to seal the nicks in the second strand during cDNA synthesis, to amplify the DNA, to detect point mutations in DNA by ligase chain reaction or ligase amplification, and also in the DNA repair. Right, so nucleases, what are nucleases? These are enzymes that hydrolyze the nucleic acid by breaking the phosphodiester bond that holds the nucleotides together. So nucleases are usually divided into endonucleases and exonucleases. So restriction enzymes are examples of endonucleases, which cuts within a DNA strand. The other part, second group of nucleases which degrades the DNA from the termini, are called the exonucleases. Now, exonucleases are enzymes that work by cleaving the nucleotides one at a time from the end. So that is why they are called exo. And its close relative is the endonuclease, which cleaves the diester bonds in the middle of a polynucleotide chain where it identifies the palindromic sequence. Now we know that these restriction enzymes identify palindromic sequences. So once when there is a palindromic sequence, it goes and cuts over there specifically. So they are very specific in nature. Now exonucleases breaks apart single-stranded DNA in a 3' to 5' direction, releasing deoxyribonucleoside 5 monophosphatase. It does not cleave DNA strands without the termini 3' OH group. That's about exonuclease 1. Then exonuclease 2 is associated with DNA polymerase 1, which contains a 5' exonuclease that clips off the RNA primer content immediately upstream from the site of the DNA synthesis in a 5' to 3' manner. Now, exonuclease 3 has 4 catalytic activities. It can go do RNase activity. It has a exodeoxyribonuclease activity, the 3' phosphatase activity, and the endonuclease activity, which is also found in its endonuclease 2. There are other types as well. Some of them requires Mg2 plus, some requires Ca2 plus for the activity. More or less, they have similar kinds of activities, just they need different kinds of molecules for triggering them, or have some special activities, like you can see the RNase activity and things like that. And also the exonuclease 4, the exonuclease 8 is 5' to 3' dimeric protein that does not require ATP or any caps or nicks in the strand. It requires free 5' OH group. Now endonucleases are enzymes that cleaves the phosphodiester bond within a polynucleotide chain, and most of them are called the restriction endonucleases. They identify 4 to 6 nucleotide long palindromic sequences, and it cuts them very specifically, leaving sticky ends, sometimes blunt ends as well. So, restriction endonucleases are divided into 3 categories: type 1, 2, and 3, depending on the mechanism of their action. Now, restriction enzymes are called the scissors of molecular genetics. So they recognize specific sequences in the DNA and breaks at certain points. So variety of restriction enzymes have been isolated and are commercially available. Like I was saying, they identify specific palindromic sites in the DNA, and they cut the uh, DNA strands. So these cuts can be staggered, which generate sticky or overhanging ends, or a blunt end, which generate the flush ends. Now, if you see that if another piece of DNA has matching overhangs, like for an carbon, the overhangs can stick together by complementary base pairing. For this reason, enzymes leaving single-stranded overhangs are said to produce the sticky ends. So sticky ends are helpful because they hold two pieces of DNA together so that they can be linked by a DNA ligase. So not all restriction enzymes produce sticky ends. Some are blunt cutters as well. For example, SMA1. Now these are the different examples of the restriction enzymes that are given over there. We need not worry about this as of now. You can check it. This is some uh, of the commonly available restriction enzymes in the website and in commercially available as well. So let's talk about some synthetases like the DNA polymerase, the reverse transcriptase. So DNA polymerase are enzymes that synthesize the DNA molecules from the deoxyribonucleotides, which are the building blocks of DNA. Now these enzymes are essential to DNA replication and usually work in pairs to create two identical DNA strands from a single original DNA molecule. Now during this process, DNA polymerase reads the existing DNA strands to create two or more strands that match the existing ones. Now, we have read about DNA polymerase before as well. So, we know that every time a cell divides, DNA polymerases are required to help duplicate the cell's DNA so that a copy of the original DNA molecule can be passed to the daughter cell. Now, in this way, genetic information is passed down from generation to generation. Now, before replication takes place, an enzyme called the helicase unwinds the DNA molecule from the tightly woven form. This opens up and unzips the double-stranded DNA to give two strands of DNA used as templates for replication. Now, Pol1 or the DNA polymerase 1 is encoded by the POLA gene and is ubiquitous among the prokaryotes. Now, Pol1 activity can be replaced by four other polymerases as well. So there are different polymerases which can take up the activity of Pol1. So polymerase 2, the primarily involved in DNA repair, and the replication restarts at the bypass lesions. So the key features is that it has 3' to 5' exonuclease activity. Its expression increases significantly from 30 to 50 to 200 to 50 copies per cell during the SOS response. So emergency response, it acts as the backup for polymerase 3 to bypass the terminal mismatches. Just a minute. Just give me a minute. So I was talking about the polymerase 3. So the primary role of polymerase 3 is it's a primary enzyme for DNA replication. So it's a complex holoenzyme consisting of three assemblies: the Pol3 core, the beta sliding clamp, the clamp loader complex. So each of them have different roles. So Pol3 core contains subunits like the alpha, which is the polymerase, eta. You have the theta, that's stabilizer. Then you have an exonuclease. Then you have a sliding clamp that ensures high processivity by tethering the enzyme to the DNA. You have the loader complex that has the seven subunit assembly that loads the sliding clamp onto the DNA. Then the polymerase 4, it's an error-prone polymerase involved in non-targeted mutagenesis and transletion, things like that. It induces by the SOS response, and it bypasses the specific DNA lesions at stalled replication forks. And finally, coming to the reverse transcriptase, it's an enzyme that functions as a RNA-dependent RNA polymerase used by the retrovirus to copy their viral RNA genome into the DNA before integration into the host cell's genome. However, it has other activities as well. So, like you can see the DNA polymerase activity. So, in nature, reverse transcriptase copies the DNA into RNA. In lab, it can transcribe both single-stranded RNA into DNA templates if a primer is present. You have the RNase H activity. So this enzyme degrades the RNA strand from a DNA RNA hybrids formed during the reverse transcription. So most enzymes comes from monomollonimurin leukemia virus, MMLV, then the avian virus, AMV. things like that. So synthesis of uh, the cDNA. So to clone genes, reverse transcriptase is used to create cDNAs, which are DNA copies of the mature RNA. So for priming, you need an oligo-DT primer, which is mixed with an mRNA. It anneals at the poly A tail of the mRNA. So reverse transcriptase is added to synthesize the first strand cDNA, and then it's basically used for real-time reverse transcriptase PCR, that's generating DNA copies from RNA before amplification via PCR, RT-PCR, and then it's used for identifying microbial diseases and cloning specific cDNA sequences. Now coming to another important part, that is about the vectors. So what is a cloning vector? So a cloning vector is a DNA molecule used as a vehicle to artificially carry foreign genetic material into another cell. So because a desired gene cannot be directly introduced into a host cell, it is inserted into a vector that can replicate autonomously. Now, the characteristics of an ideal vector. So, for a DNA molecule to act as an effective vector, it must be small in size for easier manipulation. It should be able to self replicate. Inside a host cell, it contains restriction sites. It does not interfere with the replication. When a foreign DNA is inserted, possesses marker genes to identify the correct recombinant cells, which are basically the antibiotic resistance. And they should have multiple cloning sites like the polylinkers. Some commonly used vectors includes the plasmids like PBR 322. You have cosmid vectors, the BACs, YACs, bacteriophage, lambdaphage, virus SV40, etc. Right? We are not going into the all these nomenclature stuffs like that. Okay. Now, important to know about plasmids. So that's the features of the PRB2 that's given over there. You can see the origin of replication, the Odyssey site. Then there are selectable markers. You have two markers over there. There is ampicillin resistance and the tetracycline resistance. Uh, Cloning sites, like about 20 cloning sites are present over there. Inserting the foreign DNA into specific site inactivates the gene, which allows for the insertional inactivation screening. And advantages is that it is smaller in size for easy purification, dual marker for easy selection of the recombinants, can be amplified up to 1000 to 3000 copies per cell using chloramphenicol. Cool. Okay. Now the bacteriophage lambdaphage, it consists of a head containing the viral DNA genome and a tail for infecting the E. coli. The wild type lambda virus contains a 50 kb linear double stranded DNA genome. The protein coat is discarded upon attachment to a host cell and the lambda DNA is injected into the bacteria. So that's how the structure looks like of a bacteriophage lambda one. And then that's the lambda phage DNA. So the key features is that it generates the sticky ends at the cos site. So the DNA has single-stranded overhangings that are 12 nucleotides long and complementary. We call them as the cos sequences. Then once inside a bacterial cell, these ends base pair and are sealed by ligases, turning the linear DNA into double-stranded circular molecule, and you can undergo vector modifications. That is, natural lambda DNA isn't ideal for cloning because it has too many restriction sites. So that is why it needs to be specifically designed for cloning it. Now there are two types of uh, lambda cloning vectors. One is the lambda insertion vector. So a large portion of the non-essential DNA is deleted from the phage genome. It contains a single restriction site where new foreign DNA can be inserted. And the other one is the lambda replacement vector. So they contain a stuffer fragment flanked by restriction sites, and majorly the function is when the stuffer fragment is removed and replaced by the target foreign DNA, it allows the cloning for much larger DNA fragments compared to insertion vectors. Now, replacement vectors are generally designed to carry large pieces of DNA than insertion vectors can handle it. Okay. Now we will get into the uh, lytic and the lysogenic cycles of a bacteriophage. So, bacteriophage is a virus that infects bacteria. Now, what happens in a lytic cycle? The phage acts like a hit and run parasite, eventually destroying the host cell. It starts with an entry, attachment and entry. So, the phage attaches to the bacterial surface and injects its DNA. Then, the host machinery is hijacked to produce viral DNA and proteins via replication. New phages are assembled inside the cell, and the cell bursts, releasing hundreds of new phages to infect the other cells. Whereas on the lysogenic cycle, it's a more of a stealthy pathway where the virus coexists within the host. So there is the phage DNA integrates into the bacterial chromosome, becoming a prophage. Every time the bacterium divides, it replicates the viral DNA along with its own. Now, under certain stress conditions like say UV light, the prophage can exit the bacterial chromosome and enter the lytic cycle. So okay, so the choice between a lytic and a lysogenic cycle is controlled by two mutually antagonistic regulatory genes, the CL and the CRO. So lytic state, the CRO gene dominates, repressing CL, and in the lysogenic, the CL gene dominates, suppressing the lytic genes. So genes are not essential for the lytic pathway. Often those involved in lysogeny can be removed and replaced with a foreign DNA. So the capacity up to 25 kilobase pair of foreign DNA can be inserted into the lambda genome. And recombinant phages transforms E. coli at higher efficiency using in vitro packaging systems that mimic natural viral assembly. Now, the virus SV40 vector is a spherical virus with a circular double stranded DNA chromosome. Its gene components contain small t and large t early proteins. It has the caspid proteins, the VP1, 2, and 3, and around an 80 base pair region associated with histones. Now, vector type, so SB40 is used to develop the transducing vectors, plasmid vectors, the transforming vectors. So the transducing vectors, so these vectors produce viral particles after they infecting the monkey cells to function. They contain origin of replication, transcriptional regulatory signals, including regions for splicing and polyadenylation. The late replacement vectors, so in these vectors, the region encoding the late proteins, that is VP1, 2, and 3, is replaced by a DNA insert. So, because the late genes are missing, the vectors must be used alongside a helper virus. Then, successful viral replication and cell lysis occur only in host cells infected by both the vector and the helper virus. The early replacement virus, the DNA insert replaces the early regions, specifically the large T gene. So instead of a helper virus, these vectors often use the specialized host cells like the COS cells. These are CV1 monkey kidney cells that have been engineered to contain the SB40 large T gene in their own genome. Okay, so that is a lot of information about the SB40 plasmid vectors. Let's talk a little bit about the cosmid vectors. So, what are cosmids? So, cosmids are natural essential plasmids that contain a minimum of 250 base pairs of the DNA, which includes the following sequences from the phage genome. That's the cos site you need, the sequences needing for binding of the um, terminase action to package the DNA into the empty lambda phage particles in vitro. So they contain an origin of replication, unique restriction sites, and the selectable markers like the others. Now we can talk about a little bit about the typical features of the cosmids. So they have large insert capacity. So they can clone like DNA inserts up to 40 to 50 kilobase pair, which is much larger than the standard plasmids. Now, recombinant cosmids are packaged into lambda phage heads. For successful packaging, the distance between two cos sites must be between 38 and 52 kilobase pair. They are high efficiency because they infect host cells via phage particles. Their delivery of DNA is significantly more efficient than standard plasmid transformation. And once inside a bacterial host, cosmids behave like plasmids. So they circularize and replicate independently using their own origin of replication. Now getting into the techniques which are very interesting. So polymerase chain reaction, it is one of the, or the PCR, it is one of the fundamental techniques used in genetic engineering. It occurs in three important steps like the denaturation, annealing, and the primer extension. So in denaturation, the double-stranded DNA template is heated and it opens up. So the hydrogen bonds between the strands break, separating the two into single strands. Now, what happens upon lowering down the temperature? The oligonucleotide primers binds or anneals to the complementary sequences on the single-stranded DNA. Now, these primers provide the necessary free 3' OH group for the next step. And primer extension is where the DNA polymerase activity is optimal. So you have the Taq DNA polymerase, the fusion, things like that. These enzymes add nucleotides to the primer, synthesizing a new DNA strand complementary to the template. Now, when you are done with the PCR or the polymerase chain reaction, you have to run them in gels. For that, you have the agarose gels for that. So you can run them in an agarose gel. So what happens? We know that a DNA or an RNA is negative in charge. So you run the gel from a positive to from a negative to a positive direction. So it will start migrating towards the positive end, and they will separate based on the sizes of the strand. So if it's a say 1,000 base pair runs slowly compared to a 200 base pair, it's like that. From there, you can understand whether your gene of interest has been able to amplify successfully or not. Now there are two types of hybridization that is important, like the southern and the northern hybridization. So we use the northern hybridization. Uh, so there are two ways. So the first one is that the northern hybridization is used for RNA and the southern hybridization is for DNA. So the southern blot is used for analyzing gene structure, mapping and detecting the mutations in a DNA, and RNA to study the gene expression, measuring how much mRNA is being produced. So there are different steps. So basically the common steps are that you undergo a restriction digestion, so that DNA is cut into fragments using the restriction enzymes. You perform an agarose electrophoresis, so the fragments are separated based on their size. Then you blot it. So the DNA fragments or the RNA fragments are transferred from the gel onto a solid membrane filter, like you have a nylon membrane or a nitrocellulose membrane, and you hybridize it. So the filter on which you have these nucleic acids, sorry, is incubated with a labeled probe that binds only to its matching sequence. And then when you undergo autoradiography, it is exposed to X-ray film and a dark band appears where the probe is bound. So identifying the size and the presence of the specific gene or sequence. Okay. Now we do similar kinds of experiments for the northern blotting as well, where instead of the RNA we are using the instead of the DNA we are using the RNA. For western blot, what you do, you detect proteins. So proteins from a mixture of number of proteins gives you the information about the size of the protein. So say you are expressing a protein from you have a construct, a DNA construct from that, you are expressing that in a bacterial culture and you get the protein. So you run that culture of proteins and you see where it is expressing. So what do we do over here? You run a sodium dodecyl sulfate polyacrylamide gel, that is the SDS page. You run it and then you transfer it into a PVDF or a nitrocellulose membrane, and you use a blocking reagent, say a milk powder, or sometimes we use BSA, that is bovine serum albumin, for blocking it. And after the blocking, we put a primary antibody followed by a secondary antibody tagged with a HRP conjugate or like a horseradish peroxidase. And then what we do after removing of the secondary antibody, we develop the blot using some substrate that and an enzyme as well. So upon illuminating it with under the chemiluminescence, we see some dark bands indicating, okay, so that particular antibody has bound to the protein of our interest that is present over there at that particular size. Any questions still here? Any doubts? No questions? Okay. If there is no question, then we can do some more. Well, let us talk a little bit about the cDNA libraries and the genomic DNA libraries. Okay. So, a collection of cloned DNA fragments synthesized from the RNA of a specific tissue or cell type is the cDNA library. It represents only the expressed genes, the transcriptome of an organism. So, based on the cDNA is derived from spliced mRNA, it lacks introns. So, this makes it easier to sequence and express proteins compared to the genomic DNA. Now, steps in the cDNA library construction is the isolation of mRNA, extracting total RNA from the tissue of interest, then synthesis of cDNA, creating the first and second strands of DNA using the mRNA as a template, then incorporation into a vector, so inserting the cDNA into a bacteriophage or plasmid, and cloning, inserting these vectors into the host cell. Then you have the isolation of the mRNA. So the eukaryotic mRNA has a poly A tail. So a chromatographic column contains oligo DT to catch the mRNA. When the cell extract passes through the poly A tails of mRNA, binds to the oligo DT via base pairing. Other RNAs do not bind and flow over, and you elute it in water or sometimes low salt buffer. So for that you need to synthesize the first and the second strand of DNA. So why convert mRNA to cDNA? Because mRNA is single-stranded and cannot be directly cloned or used as a substrate for the DNA ligase, and it must be converted into double-stranded DNA to be inserted into a suitable vector like a plasmid. So first, for the synthesis of the first strand, a short oligo-DT primer is attached to the poly A tail located at the 3' end of mRNA. Then you use the enzyme reverse transcriptase to which uses the mRNA as a template to create a complementary DNA strand. So it creates a cDNA mRNA hybrid molecule. And for the second strand, you need the RNA removal. So mRNA strand in the hybrid is typically degraded using RNase H, and then DNA polymerase 1 uses the remaining RNA fragment as the primers to synthesize the second DNA strand. So once the first strand of cDNA is synthesized from the mRNA, we remove the mRNA using RNase H, then self-priming happens, double stranding using reverse transcriptase occurs, and the hairpin loop is removed by the S1 nucleus to create the blunt ends. Now what you have to do, you have to incorporate it into a vector. So we use some kinds of linkers because the blunted ligations is insufficient. So what are linkers? So these are short chemically synthesized double-stranded DNA containing restriction sites. Now linkers are ligated to the blunt ends of the double-stranded cDNA using T4 DNA ligase. Now the cDNA is treated with a restriction enzyme. This cleaves the linkers to create sticky or protruding ends, making it easy to insert in the cDNA into a plasmid vector. Now coming to the cloning and the applications of the cDNA and the cDNA libraries. So basically the final stage is the cloning, where we do it using the restriction enzymes and ligase. So the recombinant vectors are then introduced into the E. coli cells, creating a library where the bacterial colony or the phage clone contains specific cDNA corresponding to the mRNA molecule. So advantages is that while both plasmids and phages are used, bacteriophage offers specific benefits like the efficiency. They are more suitable when a vast number of recombinants are needed. It helps in vivo packaging, handling, and it has wide applications. Like most importantly, it has splicing, helps in studying alternative splicing patterns across the different tissues. It since cDNAs lack introns, they can be expressed into prokaryotic systems to produce eukaryotic proteins, which is the primary goal of molecular cloning. It helps in identifying new genes and their important functions. Okay, so a little bit about the genomic library, and very short about the genomic library. So genomic libraries cover the entire organism's genome, including expressed and non-expressed genes, exons, introns, regulatory regions. So construction involves isolating and fragmenting the DNA, followed by cloning it into vectors. And then fragmentation can be physical or enzymatic, using the restriction enzymes that occur during partial digestion. So it's about the entire genome. So we use restriction enzymes for to generate the DNA cuts and introduce some blunt ends and the sticky ends, which are called the cohesive ends, as like that. Then we modify the blunt ends for cloning. We add linkers, adapters for them to join together, and we have the restriction enzymes that generate the sticky ends. Then we clone the genomic DNA into lambda replacement vectors. You have the high capacity vectors, and each of them have different functions, and some copy number might vary, some might not be able to take up a certain insert, so things like that can happen. So mainly the advantages of genomic libraries is that the identification of a clone encoding a particular gene of interest. It is useful for prokaryotic organisms having relatively small genomes. Then the genomic libraries from eukaryotic organisms are very important to study the genome sequence of a particular gene, including its regulatory sequence and its pattern of introns and exons. If we talk about the applications, it helps to determine the complete genome sequence of an organism, study and generate transgenic animals through genetic engineering, serving as a source of genomic sequence, to study the function of regulatory sequences in vitro, to study the genetic mutations, used for genome mapping, sequencing, and the assembly of clone contigs. Right? Okay. So I think that will be enough for the lecture part. Okay. If you have any, before I go to the quiz part, if you have any questions, you can uh, ask me. Any questions from the lecture? Okay, then we can start with the quiz thing. So the first question is match the following. So you can answer me in the chat box. B. Okay. So let us go from column B. So recognize a specific nucleotide sequence in DNA and cleaves the bond between two nucleotides. That is a activity of an endonuclease or a restriction endonuclease. So B1. Okay, then links together DNA strands that have double-stranded breaks. This is by a DNA ligase. So A2, bacterial extrachromosomal DNA used as a vector in molecular cloning. That is a plasmid, and enzyme required for amplifying gene of interest. That is a Taq polymerase. Okay. Now tell me which of the following are not required for PCR reaction. So for a polymerase chain reaction, which of the following is not required? Mg2 plus, DDNTPs, Taq polymerase, pair of primers, template DNA, calcium. So which are the things that you do not require for a polymerase chain reaction? So basically for a polymerase chain reaction, you require a few important things. The first thing is the DNA template that if you want to amplify. You need DNA polymerase that will help in the amplification. You need DNTPs, not DDNTPs, because DDNTPs will stop the reaction at that point. Okay, so DDNTPs are not required. You need a Taq polymerase. You need a pair of primers that will be required for the annealing, and then the extension is done by the DNA polymerase by adding of the DNTPs. You need the template DNA, and you need a buffer that has magnesium. So you don't require calcium, you don't require DDNTPs. So 2 and 6, that is B. Okay, the next one. Suppose you are working on gene A and you need to amplify the gene for cloning, that is in a plasmid vector, vector P. If the initial number of DNA molecules at the start of a PCR reaction is 10 to the power 6, what will be the number of DNA molecules after 30 cycles? So how will you do it? So first you need to find the number of DNA molecules after a PCR reaction. So let us do it in this manner. So you have the final number of DNA molecules will be equal to initial number of DNA molecules into 2 to the power n, where n is number of cycles. So if you start calculating 2 to the power n, that is 2 to the power of 30. That is equivalent to around 10 to the power 9. So if it is around 10 to the power 9, then what you get 10 to the power 9 into the number of initially present molecules. So that is 10 to the power 6. So the right answer is A, 10 to the power 50. Okay. Question 4. Which of the following processes are involved in cloning whole animals? So if you want to clone whole animals, what would you require? Is it in vitro fertilization, embryo splitting, somatic cell nuclear transfer, or things like that? So, in vitro fertilization is the method of sexual reproduction where an egg and sperm are combined in a lab. It results in a genetically unique individual, not a clone. Embryo splitting, it's a form of artificial cloning. So it mimics the natural process that creates identical twins by splitting a single stage embryo into multiple embryos, each of which is a clone of the other. Somatic cell nuclear transfer, it is a primary method for reproductive cloning. That is Dolly the sheep. You have heard of Dolly the sheep, so it is similar to that. It involves transferring the nucleus from a somatic body cell into a enucleated egg cell. And gametic cell nuclear transfer, this is not a standard process used for cloning whole animals, as gametes only contain half of the required genetic material. So the correct answer over here is you can do it for 1, 2, and 3. So ideally the correct answer should be 2 and 3, but I don't know why the option is not there. So anyways, the correct answer is 2 and 3, but in vitro fertilization it's not a cloning thing, so better not to give. So 2 and 3 is correct. The next one, which of the following is the first genetically engineered pharmaceutical product? First genetically engineered pharmaceutical product. No, it is option D, human insulin. This was first genetically engineered the product for human use, developed by a biotech company Genentech and manufactured by Eli Lilly and Company. So it received the approval in 1982. So scientists used DT or the recombinant DNA technology to insert the human insulin gene into the bacterium E. coli, which is then produced the insulin protein, and it was a major advancement in the field of RDT. Okay, now coming to the next one, which says, which of the following statements are part of the process of Sanger sequencing? So this is one of the sequencing techniques. So Sanger sequencing is also known as chain termination method. So Sanger sequencing relies on dideoxynucleotide triphosphatases, the DDNTPs. These lack a 3' OH group, which prevents the formation of phosphodiester bond within the next incoming nucleotide. So this is correct. So analysis of fragments by gel electrophoresis, yes, that's fine because the resulting DNA fragments of varying lengths are separated and it helps you to determine the sequence. Chain termination by incorporation of DNTPs, this is wrong because we have already used the DDNTPs. And chain termination by cyclic NTPs. So cyclic nucleotides are not used in this particular process. So only 1 and 2 are correct. That is option A. Which of the following is essential for the action of DNA ligase? So for the action of DNA ligase, what is the one that is actually required? Very direct. It's an ATP, very good. So it's an ATP molecule that you require. Okay. Now the next one, which of the following is true for cloning of a mammalian protein coding gene? So if you want to code for a clone of a mammalian protein, what do you do? Yes, so the genomic DNA contains introns which would prevent proper protein synthesis in the bacterial host. So the first one is incorrect. And CND, that is mRNA or the total RNA, cannot be directly used as a template for the PCR amplification process or the direct insertion into the DNA vectors. It must be first converted to a DNA, a cDNA, and then it is used as a template for amplification of the gene of interest. So option B is correct. And the last two is, which of the following properties of plasmid DNA are required for cloning and expression of protein coding genes? Which of the following properties of plasmid DNA are required for cloning and expression of protein coding genes? So, to successfully clone and express a gene, the plasmid needs an origin of replication to replicate within a host, MCS containing restriction sites for inserting the gene, and promoter terminator regions to drive the or stop the transcription of the gene into mRNA. So we need all of the above, right? And last one says agarose gel electrophoresis separates DNA molecules based on their molecular charge, molecular mass, concentration, GC content. No, that is not right. So in agarose gel electrophoresis, all the DNA molecules have uniform negative charge to mass ratio. So therefore, they all move towards the positive electrode. So the gel acts as a molecular sieve, allowing smaller DNA molecules to move faster and further through the pores than the larger fragments. Thus, we separate it based on size or mass. So it is molecular mass. That is option B. Okay, right then. Um, no, nothing else. If you have any doubts from this class or the previous class or any assignment related problems, you can ask me. Okay, then. Thank you very much for joining. We will meet. Okay, so there is an extra session on the 7th of April where we can discuss some doubts, some quiz questions, some maybe some important lectures here and there. We can talk about that. Okay, that will be a 2 hour class, extra session for some special doubt clearing. If you have any questions from the other assignments, from because the exams are coming up, so there can be some doubt. So it's more or less like a doubt clearing session more than any week wise lecture. So feel free to join on the 7th from 6 o'clock, right? Okay, then. Fine. Thank you for joining. Have a good day.